So today we're here to discuss sustainable practices and how we can green our labs. Because as scientists, we have to lead by example, I think, and we can discuss how with a few changes in our everyday routines in the labs, we can make a difference and reduce our carbon footprint and maybe even more than that. So I'm passing on to Megan. Megan.
Hi, everyone. And again, yes, thank you all for being here today. So we're very excited to host Dr. Federico Forneris and one of the original Meshwork founders, Dr. Julie DiMartino. Dr. Forneris is a full professor at the University of Pavia in Italy at the Laboratory of Structural Biology. He received his master's degree in physical chemistry from the University of Turin and his PhD in basic and applied biomolecular science from the University of Pavia. He did his postdoctoral work both at the University of Pavia and the University of Utrecht in protein structural biology and crystal and structural chemistry, so lots of great background and insight there. Dr. DiMartino has been an assistant professor of cell biology and anatomy at New York Medical College for the past 2 years. She received her bachelor's degree in cell biology and physiology, her master's degree in cellular and molecular genetics, and her PhD in cellular biology and pathophysiology, all from the University of Bordeaux in France. She did her postdoctoral work at the Icahn School of Medicine at Mount Sinai Hospital in New York, and she's very passionate about running a green lab, and she has lots of great insight to offer today. We also just wanted to highlight for any new people what the Meshwork is and what we're doing here. So we're an online network that aims to connect researchers, and in particular early career researchers, from across the globe who study extracellular matrix. We offer online talks such as this one. We highlight early career researchers and resources in our monthly newsletter, and have been organizing a series of recorded interviews that you can find on our YouTube, as well as this talk that you'll see there as well. If you have any suggestions or anything you'd like to see, please contact us. We always love to have your input. And with that, I can let our speakers take the floor.
So in the next 15 minutes or so, I'm going to try to go over the different practices that I implemented in my lab as I was starting, like as Megan said, 2 years ago already. I first want to thank Chloe Young for suggesting us to do this event because that's very close to my value. Usually people take the angle to say, well, there is no planet B, so we must, this is our duty to increase our sustainability in the wet labs so we can reduce the environmental impact of our work and improve the workplace safety. But one thing I would like to start stressing out is that those practices also save money. And I don't know if you have seen the trend in the couple past decades or so, but unless you're working in China, most of our government are not so much interested in investing in research and development. So that's going to force us to try to do more with less. So in the lab, we have 4 different strategies that we use to try to work toward having like more greener practices, which are changing habits, recycling, reuse, and upcycle. So I'm going to go over a few examples of each of those strategies that we have, because what I found out when I started talking about this is that most of the time, it's not that people are not willing to do those changes, is that they don't exactly know where to start or what they can do or cannot do without impacting their results. So changing habits. So the first thing is that very easy. In the lab, we don't leave anything on, light, machines. That is like very simple. Turn off everything when the last person leaves. That saves energy. When it comes to lunchtime, what we also have is that I provide a reusable diningware, so we don't use any single-use plastic to have lunch together. Another thing that we do is, so this, I know that not everybody can do that based on the level of safety of your labs, but we add a lot of plants to our lab to clean the air of potential chemical leak that could be occurring during our experiments. Another change of habit is to buy non-electrical tool, but also efficient machine that you don't need to keep on all day. So I don't know if you all have that old dry bath that you have to leave on forever because it takes forever to go to 95 degrees. And it's also a pain to find the right temperature and setup. Well, you can easily replace that by a brand new machine that's like less than $200 that actually is going to go to 95 degrees very precisely in about 5 minutes, warm up your sample, and then you can just turn it off until the next users use the machine. So remember to try to buy efficient, energy efficient machines. Another thing that you can do to change your habit is to buy bulk and not racked. So most of the rack, although some companies are starting to make cardboard racks, most of them are not easy to recycle. So buying bulk helps you to decrease the amount of packaging around your items that you need. We also use that strategy in tissue culture, where you all know that we have to work in sterile condition. We buy bulk, sterile, non-single wrap pipettes, as you can, I don't know if you, yeah, as you can see here, instead of having those single wrap pipette. By doing so, we decrease the amount of waste in tissue culture. Another thing you can do at the bench is to use non-sterile glass pipettes. So if you work at the bench and you don't need to be in sterile condition, those pipettes can be used and washed and dry, and they even can be autoclaved if you want them to be super clean for your next experiment. And another habit change that you can do is to, instead of buying things that are pre-made, make them yourself. So for instance, in my lab, we use a lot of those small chambers to do live imaging. They cost a tremendous amount of money. So what I did is that I purchased a 3D printer. I spent some time trying to model, let me remove that, the design that I wanted. So for that, well, it takes a little bit of time, but then at the end, you have your design. Your design is actually working, holding liquids. This resin is autoclavable, so you can actually have it in sterile condition to use in TC. And then you can go beyond, like we needed longer agarose gel, so we just printed out the agarose gel mold that we exactly wanted, the number of wells you want. So that is a way to, so you save the shipping, so you decrease the carbon footprint, but you also have the exact design of what you want to use in the lab. The next strategy is to recycle. So we really recycle every single item that can be recycled. Anything that didn't touch biohazard and that a material that can be recycled goes to the blue bin. But another way that you can recycle is to be a scavenger. People like toss a lot of valuable things, or when lab retire, they shut down, and usually everything goes to the trash. So this is a good way to recycle whatever is already there, to save money and save a little bit of carbon footprint. Another strategy we have is to reuse. So we have a lot of glassware, and we buy glassware over plasticware because they have a long lifetime. And we just autoclave what we need to be sterile. We don't autoclave everything that is glass. If it's something that we don't need sterile, we just don't autoclave those, because autoclaving also consumes energy. Clean and reuse tubes in non-sterile condition. Don't buy them racked unless you need the rack. So at the beginning, I bought some rack tube because I wanted those plastic racks. But then now we use in bulk, and we just wash them, put them in buckets that we reuse when we need. And you have to know that those tubes can also be autoclaved. So you can also autoclave them if you want. Clean and re-autoclave like glass. So for example, in tissue culture, we use those pasta pipette to aspirate with our vacuum system. We just like collect them in bleach and then clean them with water and autoclave them back. And we have this circle where we reuse them over and over until they break. Another thing that we do is that we buy our tips sterile and filter for tissue culture, but then we recycle those boxes and buy the non-filter tips for the bench and just re-rack those boxes to save plastic boxes. And we also reuse those boxes for various things, like things that we need to store. The 1000 tips are great for that. If you're using repeaters that use special tips, you can recycle them and reuse them over and over. So we just write on the tip what we pipette with, and we dedicate it for it and keep reusing those tips. Another thing that we reuse is OCT mold. If you guys are doing tissue embedding in OCT, once they're frozen, you can store them in little plastic bags and recycle those molds over and over. Another way you can decrease the amount of plastic is to buy a large quantity of concentrated solution and dilute in glass container. So that's what we do for PBS, for tissue culture. We actually prepare the 10x PBS, diluted in those dedicated bottle for PBS, but we also do it for trypsin, for instance. We buy it concentrated and we dilute it in those glass containers. Another thing you can do to save plastic is to make media from scratch. So basically, so we kept the color coding of the Gitco, so it will not be confusing for our students when they move to a lab that use those already pre-made media. But basically, we just autoclave water in there. We have space in our lab where we save, where we put the empty bottle, the bottle with water. And then this is how 10 liter of media look like in my lab. So that's also very efficient in terms of storing in the cold room. So one thing that we also do, and that's going to help me transition to the next step, is to upcycle. So once we're done with this bottle of powder, media powder, we reuse it, for example, to hold our pen in the lab. So upcycling is the last category I'm going to talk about today. It's repurposing things for other purposes. So one thing we upcycle is our tomato sauce. So we basically clean the jar and then use them to autoclave tips because they are autoclavable. Another thing that we upcycle is when we receive cold item in the lab, we recycle the styrofoam box. We scavenge in the hallway people that are tossing those tube racks, and then we make freezing boxes to freeze our viral cells in -80. Remember our bulk pipette in those plastic bags. So it happened that those plastic bags are thermo sealable. So if you have a sealer, you can actually use it to seal and save Western blot membrane, for instance, and save them. So this is zero trash. So we use the pipette and then we use the container that we bought the pipette in. So with that, I'm going to put it all together because what I found out is that people doing tissue culture in sterile condition are very scared of getting contaminant and so on. So I have to disclose that we don't use antibiotics in the lab. Most of our tissue culture is done with antibiotics, and we don't get a lot of contamination. And that's how the setup look like. So we have our pipette in the bags under the hood. We have all our tips and so on, our media that we make ourselves. And then on the right side, we have bin to collect the pasteur pipettes that we then reuse and recycle. So with that, that's going to be it for me. I just want to thank you guys for your attention. And I also want to tell you that you should trust your crazy idea, but most importantly, find the people that are crazy enough to follow them. And I just put here my Twitter handle because when I have a little bit of time, I tweet about those different strategies that we use. Thanks for attention, guys.
Thanks a lot, Julie, for this amazing presentation. Very nice ideas thrown in there that we can discuss later. And now let's pass on to Federico for his presentation.
All right, thanks. So I would like to catch the opportunity of some of the suggestions provided by Julie to show proudly my pen holder, which is actually the upscaling of a container that was originally used by a glow discharge system. So I'm actually, let's say, focusing on this type of activities as well. And another thing before I start that came to my mind that we often use, even without thinking, is that since we established my lab 10 years ago, we've been recycling computers. So we have basically never purchased a new computer for instrumentation because we've been recycling computers that were discarded by others because they were not sufficiently performing. That said, let's start with the story that I want to tell you. So I hope that you can see my screen. Right. So the story that I'm going to tell you today is actually something that emerges from a series of ideas that came out based on what we normally do in the lab. So, I mean, as some of you know, we are actually a structural biology group, and I thank the Meshwork for the introduction, actually. That was very pleasant to hear. We have been studying collagen and collagen post-translational modifications quite extensively over the past years. And actually, the focus of my lab is also extending towards other extracellular systems, including synapse assemblies in the neuromuscular junctions and in the retina, as well as some signaling cascades that involve also some very annoying insects, including mosquitoes. So the way we approach all these systems is very similar. I mean, although they are very different. We basically carry out a number of in vitro studies where the central activity, I hope that you can see my pointer, is actually related to obtaining samples, mostly recombinant samples, that we can then use for a large variety of studies, going from stimulating cells into structural studies, biophysics, biochemistry, and so on and so forth. And to produce all these samples, we actually established a number of assets. And in particular, I'm very proud to say that in Italy, we were amongst the first who managed to set up a lab for large-scale recombinant production of proteins, of extracellular proteins, using mammalian cells, and in particular, human cell cultures, HEC2 trees. This is where our consideration regarding production of lab waste goes. First of all, I would like to emphasize the fact that the bottles that you can see inside the incubator are entirely coming from recycling. I mean, we are actually reusing those bottles many, many times. We did some efforts in finding which bottles could be well suited for this type of shaking cultures. And I mean, we did an initial investment that was quite considerate, but afterwards, I mean, these bottles are with us since many years. But the additional point that I would like to address today is that other than plastics, other than equipment, other than the various lab resources, there is a source of waste when you do cell cultures that is massive. That is, you normally focus on running your cultures, for example, for producing recombinant proteins, because your aim is to obtain research results and possibly bioproducts to share with collaborators. But in the end, the massive amount of liquid waste that you produce is something that typically goes unnoticed. We trash a lot of exhausted cell culture media, and we do it routinely. And in most labs, the way we dispose of these materials is simply adding bleach, letting the treatment go, and then possibly either trash it via some, let's say, official discarding pipelines, or drop it in the sink, which is definitely not something that goes well with the environment, as well as it produces an insane amount of liquid waste around. So one day, I was actually discussing with a collaborator that came into the lab, that is Alberta, who actually became my partner in crime in the project and also my partner in life. And well, by discussing with Alberta, who's actually a plant physiologist, we started discussing the possibility of harnessing some of the systems she's been studying for quite some time that are microalgae for the purpose of recycling exhausted cell culture media. The pictures that you see here are examples of some of these wonderful vegetable systems that are normally exploited for production of organic molecules, for production of lipids, as you can see from the example here, even as food additives. I mean, this is spirulina, the famous spirulina microalgae that is nowadays considered a superfood, okay? So Alberta introduced me to the concept of bioremediation that is nowadays becoming a trend in sustainable practice that makes extensive usage of microalgae to clean up mostly wastewaters coming from a variety of sources. And I mean, by discussing with her, I actually learned that microalgae can process agricultural wastewaters, urban wastewaters, even industrial, heavily polluted wastewaters containing, for example, heavy metals or other organic molecules that are incredibly hard to clean up. And the advantage of these processes is that if you manage to establish the right pipelines to process these wastewaters with microalgae, you end up obtaining water that can be reused because it's been decontaminated, but also you obtain biomass that can be achieved from the microalgae that can in turn become a very reliable source for organic molecules. Okay, so these are the typical industrial pipelines that are exploited with microalgae for bioremediation. And obviously, I mean, we could not start with something like this in the lab scale, but we started thinking, well, what if instead of using the conventional wastewater sources, we start with our wastewater sources, our exhausted cell culture media, and we transform it, or we attempt to transform it into something where we can grow microalgae for research purposes, and possibly also obtain something from those microalgae that can later on be used for other purposes. And actually, well, at the beginning we thought, okay, maybe this is too crazy, maybe this is not going to work. But I mean, we wanted to give it a try. This was a Friday afternoon idea, as it typically happens, by the way. So after a few weeks, we started thinking about how to transform this idea into a pipeline, okay? And the pipeline was basically including collection of the exhausted cell culture media, starting from our lab. We produce a lot of exhausted cell culture media every week. Processing, so transforming the exhausted cell culture media into something that can be stored and that doesn't represent a source of concern in terms of biosafety. Reformulate this material into something that can be used for growing microalgae, and then attempting to grow microalgae inside and analyze the results, okay? And before showing you some of the results, I would like to anticipate that this is actually very successful and is working very well, to the point that I'm not going to disclose anything about how we process and reformulate the material because there's a patent application pending as of now related to this topic. But basically, the idea that we wanted to establish was, well, what if we can collect these exhausted cell culture media, transform it into something where we can grow microalgae, and then compare it with this BG11, that is the standard medium that is used worldwide for growing microalgae, and seeing whether these algae can grow first, and second, how they look like, and possibly also if they do something that can be useful, okay? And in the animation that I was showing here, you can see a number of different microalgal strains growing in this homemade shaker that is currently hosting some of our attempts. And you see the different colors that represent also the different types of algae that are currently being grown. So thanks to the efforts of the heroic postdoc of Alberta's lab, Simone, we actually started to, let's say, develop some of these formulations. And to make a long story short, it works. And it works like a charm, to the point that if we just look at how much these microalgae work and grow inside the formulations that we make, well, we actually see that what we provide in terms of, let's say, resources like sugars, lipids, amino acids, all the necessary ingredients for biomass growth that is contained in our exhausted cell culture media is actually doing very well to the microalgae. Here you can see the comparison. I mean, I think that this visual comparison here is probably the best thing that you can see. Obviously, the darker means that you get the largest amount of algae growing in the system. And you see that these are different formulations where we mix and match different sources of exhausted cell culture media that we reprocessed in the lab. And basically, you see that it's actually working. It's actually working very well in producing more biomass. And we are currently in the process of analyzing what these microalgae are producing and how we can possibly extract some useful materials out of these algae as well. These experiments were carried out in relatively small scale, so moving from these 24 well plates into these small flasks. But actually, the nice thing that we noticed as well is that the process is very well scalable. This is an attempt that was done with a 30-liter photobioreactor, and you see that the results are very similar. You see that with BG11, where obviously the performance is very nice in this type of bioreactors, you get some very nice growth, but when it goes into our reformulated media made using exhausted cell culture media, you see how dark this gets. And you can imagine that the amount of biomass in here is actually quite remarkable. And I mean, Alberta's lab that is focusing on this material, they're very happy about what they are obtaining because they would have made much, much larger growth to achieve this type of amounts. And in terms of scalability, the next step is actually to move into 100-liter photobioreactors that are currently being implemented in our institute to seeing whether this could possibly achieve a semi-industrial scale for processing this material, not exclusively from my lab, but actually also from other sources. And regarding this, well, the conclusion is obviously that our trash is actually becoming very useful for others. And this is something that, let's say, opened up our minds. And regarding this opening up the mind, we decided to do a little survey. So we sent a questionnaire to some of our colleagues here at the University of Pavia, asking them how much exhausted cell culture medium they produce on a monthly basis, and what is their problems, what could be their problems in terms of disposing these materials. We figured out that, I mean, the current availability, just by looking in our surroundings, is at least 20 liters per month, which means we can fill one of these bioreactors per week quite easily, just by looking at the material that I produce plus the materials produced by others. And we can go to something that approaches industrial scale if we look at what can be obtained in our institution. I wish to point out that these estimates do not take into account any exhausted cell culture media that contain antibiotics or that can be sources of concerns regarding biosafety. So we're talking about materials that, similar to those that we produce in my lab, are completely, let's say, safe and can be reused directly without any additional concern. And considering a possible cost versus benefit balance, well, in our questionnaire, we also asked our collaborators and our colleagues here regarding how much they spend usually for disposing of this material. And we noticed that, I mean, there's not that much of an expense, but actually this is not zero. And so they will be very happy in passing these exhausted materials to us for this type of repurposing. And next to this, we started thinking about how this can become something also at a larger scale, so not only restricted to our university campus. The microalgae market is exploding. There's a forecast in 10 years that says that this will possibly duplicate or get even bigger. And this does not take into account the possibility of using microalgae for nutritional purposes. So we're just looking at the usage of this type of materials for non-human nutrition and also for obtaining, for example, organic molecules for pharmaceutical purposes or for research. And next to this, the amount of exhausted cell culture media is also going to grow massively. And we all know about this because we are all aware about how much of this material we produce on a daily basis. So in this respect, there's an opportunity to transform this not only into a sustainable practice, but also into a profitable business to a certain extent. And this is actually one of the directions that we're taking into consideration at the moment. So summing up, basically, what we are currently considering is to transform this vertical scenario where you go from cell culture into your research products, into liquid waste that should be disposed, into what we consider a circular scenario, where through this reformulation, collection, and reusage for a completely different purpose that is related to studying and exploiting microalgae, we can obtain more research results, we can obtain more bioproducts, and we can get rid efficiently of something that can be a concern regarding environmental pollution. With this, I finish, and I hope that I stimulated some additional ideas and perspectives into, let's say, practices that are related to different labs. Perhaps one day we may get in touch trying to find ways to collect also your exhausted cell culture media, who knows? And well, it's time to thank some people. So obviously, Alberta and Simone for their brilliant capabilities in handling these organisms that are definitely not my core business in the lab. My team, so those who produce the waste, so the main raw materials for the purpose, and also, let's say, the funding, because actually this idea, other than becoming a patent application, has also been funded through one of these NODES initiatives very recently. The PI is Alberta, and I'm joining, and I'm grateful of being part of this team. And obviously, all the, let's say, funding agencies that are supporting my lab and are allowing us to do the research that allows us to produce the raw materials for this second life process. Thank you very much.
Thanks a lot for your presentation. Amazing ideas. And very stimulating and like food for thought, I think, for all of us about what we can do with the liquid waste that we produce. And it is a lot, and you're right about this. So we are taking questions. You can use the Q&A function or just like raise your hand, whatever you want, whatever works best. And maybe I can start by asking one question, and that goes to maybe both of you, since you are both using sustainable practices in your labs. If you have a rough estimation on how much money can labs save per year, let's say, approximately, if they follow more sustainable practices.
I don't have that on my hand right now, but I have a plan because I was hired at the same time as other junior faculty that don't use those methods. So I want to wait a little bit more time, but try to compare roughly how much money they spend on their plastic compared to mine, for example. Because I can tell you that when I go in the hallway and I see how much they toss, we have a very like lighted bin compared to them. So I am pretty sure I'm saving a lot of money using those practices. Yeah.
Yeah, if I can add, I also don't have, let's say, estimates, not even rough estimates regarding what could be the impact, other than, for example, the saving related to the costs associated to disposing of the exhausted cell culture media in our case. But what I can say is that for labs, for our partnering labs, those who work with microalgae, there's, let's say, a lot of gain because they basically obtain all the media they need without any cost. So in this respect, the benefit is something that translates other than the unit that is, let's say, adopting the sustainable practice into the extended unit.
Okay, thanks a lot for your answers. I think maybe the Q&A was off, but now it's on. But anyone who wants to submit a question, they can also just raise their hand.
I have a question for Federico.
Yeah, perfect.
Do you think that this practice is something that you could translate into a machine that you sell to institute, so then now every institute can recycle their media at their scale? Is that something that could be, envisioned with your technique?
In a long-term perspective, I think that this could be part of the plan. Meaning, obviously, among the various things that you can do with the microalgae that you grow in exhausted cell culture media, there is also the possibility of transforming them into raw materials that can be used as boosters for growth of other cell types. I mean, this is something that has been published by some groups in last year. And in this respect, we're actually taking this into consideration to further circularize the process. So in this respect, I mean, now we are at a very early stage where our main focus, for example, is to make sure that what is collected in terms of exhausted cell culture media is properly kept, stored, reformulated. So, for example, we're implementing some digital systems to track basically every ml of the materials that get transformed, so that we can get a perfect tracking of what happens to the microalgae that then are processed later on. But yeah, I would say, why not?
That would be super interesting.
Megan, you have a question?
So this question is for both of you, but did it take a while to get these practices to work in your labs? And what was really the hardest part about making these changes and being able to keep them consistently as you're bringing in new students?
So I can start. So for me, it's pretty easy because I started from scratch. And I always say, I think it's easier to have that line of thinking before you start your lab, because then, it becomes the culture. So, when students get to the lab, they see that this is our pipeline, this is how we do things. So it's very easy. I think it's more challenging when you have a well-established lab and you all of a sudden want to implement all those. And what I will personally recommend is to do one change at a time, when you want to transition. But I really think, like, if you want to have your own lab, if you think those pipeline ahead of time and do, like, I mean, I try some of these during my postdoc to see if that will work. I was like, oh, that works. So I will use that, you see. So I think that is easier when you start from the very, very first day. And the thing you will need, though, it's a little bit of space, because we need bucket to contain all our used tubes. You need a space to have, all the glassware. So you need a little bit of space to have all that. You cannot use like a tissue culture hood that's too small, because you see the amount of things we put under. So we need at least like a 4-foot long tissue culture hood, so we can have everything underneath and still have space to do the tissue culture, for example. But yes, doing it step by step when you want to transition your lab, I will recommend that. But if you start starting from scratch, it's very easy. Yeah, I would say.
Yeah, I cannot agree more with what Julie has just said, because for us, for example, the implementation of these pipelines was relatively easy in terms of our individual lab, because we said, okay, let's give it a go, let's try it. We were all willing about doing this. So changing the habit was fine. But for example, when we approached some colleagues in the institute who were used to do some other practices, we noticed that even collecting a few mLs of their exhausted cell culture media was not exactly straightforward. So in terms of, let's say, leveraging on the fact that a new lab that establishes new pipelines can actually adopt these practices in a much easier way, I cannot agree more.
All right. A question from our Q&A for Julie, and maybe Federico also can answer. What singular sustainable practice do you think has made the greatest impact in your lab? Whether that was financial or just to make overall your lab more sustainable?
I think making media from powder, it's probably the most efficient in terms of cost saving. I made a calculation for that, and it's actually like the bottle of media is 30 times cheaper. The bottle of 30 times. So when you think that we do a lot of tissue culture, we go through a lot of tissue culture media. So yeah, that has a huge impact financially. Diluting trypsin, it's 16 times cheaper than buying it already prepared. So, you know, and if you think, so your original investment is to buy bottle that can be autoclaved, but once you have like invested in that, I think 10 liter of DMAM, it's about $20. So compared to when, and also you have to think beyond just the cost. It's like, I can store 200 liter of media in a shoebox because they are very small and compact, right? And then we just prep them. So I didn't went through the way we prep it, but we prep them by batch of 4 liters. So we actually then make them like 8 bottles at a time. So it's not like a huge burden on the people in the lab to make the media. Like, you know, I also think that when you implement this type of practice, you have to think about how much time it takes to actually do this. So I usually use the rule of like, can I do it myself? If I can do it, then I'm like, okay, people in the lab can do it. So then it's not a burden. Like if you have to dedicate 1 person to make the media, then I don't think it's useful. But to be honest, it takes us about 30 minutes per month to prep our media. And that has a huge impact financially. And that is like 100%. And I just want also to let you know that, you know, all the bottles we have, they are like actually 100% reusable, because the label we have are autoclavable. So we don't even have to change the label. So I mean, I saw this thing through that you just have to wash the bottle, autoclave, and we also, in terms of autoclaving, we don't have a system where someone autoclave for us, but we created something with the lab around that when someone has to autoclave, everybody gave this stuff. So we always run 100% full autoclave because we know how much energy it takes to sterilize things. So we try to also like work on that.
Okay, nice. Yeah, nice idea. I think to do it this way, and indeed, I think it doesn't take like too much time. And I think it's the same like for the PBS, it should be also the same. That doesn't take too much time.
Well, we do all the solution from scratch. So we don't buy any plastic bottle, like any solution in the lab, name it. We have powder and we make it in glass bottles.
Okay. Dimitra, you have a question?
Yes, hi. Thank you so much for those great tips and ideas. Actually, it's not a question. I was impressed by this practical idea about the 3D printer, and I was wondering if there are any further comments that you can share, Julie, about that.
The 3D printer, so it's
Yeah.
like $250 out of Amazon, basically.
Okay.
And I use Tinkercad. It's online to make the designs. And it's pretty straightforward. I'm not like a 3D designer, and I was able to get along with printing a lot of things. So it's and that was like a lot of try and error. But for example, I found out by autoclaving it that you can autoclave that resin. So what we do with those chambers is that we stick a glass bottom to it. So that we found that we have to autoclave the glass separately, and then we have to bond the glass under the tissue culture hood with it's UV activated resin. So you use like, we basically use like, you know, the nail UV lamp. So we put that under the hood, and that seal the glass underneath, and that you have then like a completely sterile system where you can grow cells and do movies and whatever you want to do. But what is great with that is that you have as limit only like your imagination. Like, let's say you want 3, or you want like something very specific to fit a microscope or something, then you can just 3D print it. So far we have 3D printed those designs. We have made those agarose gel casts that are a little bit longer, but with the exact number of well that we want. Those are the main things we did. Well, we printed some Pokemon to hold our tubes, but that was more fun than, you know, make tube holders that because you had the design online. But for example, I recycle. So a lab recently closed, and they have those rack for -80s, you know, that are very expensive. But they are the one that needs to have spacers. So it's a little piece of plastic that you put to hold the boxes. But those spacers over time, they disappeared. So I just 3D printed them. So you know, like it probably cost me like $5 of resin. And I saved the cost of a rack. That a -80 rack is probably $150. So yeah, so it's just like I try to tell my students, just think beyond what's possible and just make it happen, basically. Because with the 3D printer and the resin specifically, you can do whatever you want. It's actually it can hold liquid. So, you know, as long as it can hold liquid, you have no limitation.
Yeah, that sounds fantastic.
Yeah. And on
Yes.
that note, people are asking if your 3D printed designs are open access.
I can share. I mean, I don't mind sharing them. That you know, anyone that want. But to be really honest, it's very easy to design. Like, you can really design it exactly the way you want. So I made my chamber that they clip exactly the size of my microscope, so it's sealed tight, and I can do live with it.
Oh, okay. So basically, you also don't need to buy the EBD's lights anymore.
No, I don't anymore. But don't tell EBD that.
Yeah, for sure.
Well, but
it will be a secret.
Just for disclosure, they had very nice template telling you how big are each of their designs. So that's very useful when you design yours.
Yeah, so you put everything in the 3D printer and But how expensive, for example, is to obtain the 3D printer and all the materials?
So the 3D printer by itself was like $250 out of Amazon. It's a very simple one.
Oh, okay.
And then, like, for example, the cost of one of these slides, it's about 15 cents.
Okay.
So usually when you buy it, roughly it's like $4 to $5. So just so you know. Then on the 15 cents, so 15 cents, you add like the price of your cover slip. But you know, I think that's pretty decent saving.
Yeah.
Knowing that you have something custom that really fits your needs and your machine.
Yeah, for sure. And also you can, I think, print less, for example, wells if you want. Then you don't have to spend the whole thing with 8 wells, for example, just to use.
If you want a bigger surface, for example, like, and you want to print only 3 wells because you have 3 conditions, yeah, you know, it's endless possibilities.
Yeah. And Federico want to ask something?
If I can add a little comment on 3D printing, because we also made our own journey into 3D printing, and actually we made some labware as well. So I second completely the suggestions from Julie. We also use it for teaching, for example. So this is something that we made with our own little 150 euros 3D printer. And so the possibilities are endless, and it's extremely versatile, because sometimes you find in trouble obtaining the right tools. For example, we made our own customized racks for the Biorad NGC, the chromatography system, because we could not find on the market exactly what we were looking for. So we made it. And sometimes is incredibly helpful, and is also very, very cheap. So I suggest everyone to take this into consideration.
Totally. And if you need to, so the resin one has to be cured with UV light right after that. One great source of UV light are those old UV tables that people use to cut gels. That works perfectly to finish the curing of the resin. So, and it doesn't take a lot of time. Yeah.
Very nice idea. And another question we have from the Q&A. If you are aware of any regulations or pushbacks that surrounding buying or reselling old lab equipment.
Well, maybe I can start replying here. So, I mean, I think that it's entirely dependent on the countries and also on the individual institutes. For example, here in Pavia and in general in Italian public universities, it is almost impossible to resell used materials, in particularly equipment, because there are some very complicated inventory processes. And also, I mean, also focusing on what we discussed today, it is very unlikely that labs will, let's say, cease using their equipment unless the equipment is really meant for its final destination. The opposite instead is related to possibilities of buying used or refurbished materials. So this is something that I've been exploiting in the past, and I actually found it incredibly useful. Again, I'm touching the topic of, let's say, computing equipment, because that is what, in my case, was offering the best opportunities. But for example, when it came to equip the computing labs with new monitors and also with some additional tools like keyboards, mice, and so on, I mean, we bought 100% refurbished materials from companies that could sell to university labs without any problems. We saved a lot of money, and actually we got probably tools that we could not afford if they were completely new. So in this respect, this is something that I strongly encourage everyone to take into consideration, and that goes well also with more specialized equipment. Very often, when you get demo on site for equipment, you get also chances to negotiate for very effective purchase of the demo equipment rather than the new equipment that is normally sold by the companies. And this is something that perhaps may end up with the perfect match between the seller and the buyer.
Yeah, here in the US, there is specific companies where you can buy refurbish. So they clean, disinfect, you know, like all the material, they test it out. So there is a lot of like opportunity for microscopes that you can have refurbished rather than having them brand new, and that will save you like a great deal of money. And then, I mean, I just want to strengthen it again that, you know, within your own institution, as lab starts, some labs end, and reusing material from those labs that retire is very efficient way to save money, because most of those equipment work, and you just have to move them from one room to another, which is very, very convenient and very easy. I didn't purchase a minus 80. I got a minus 80 from a lab that closed. My minus 80 is probably 40 years old and never gonna break, because 40 years ago, they were doing very good machines.
Yeah.
So, you know, I can leave it open
That's
for
true.
15 minutes. It doesn't go down in temperature, right? But I think those are things that save energy along the way, because you don't buy new, you don't get this thing transported to your lab and so on. So yeah, thinking about what's on site. And those are things when you interview, you can ask, is there any material that, you know, are gonna be available because of lab retiring? And then looking into the trash, people like, I have Vortex that works perfectly that people just tossed. I have shakers that, you know, you just have to change one piece and they work. So, you know, just have to MacGyver the thing a little bit, you know, and repair.
Yeah, I think lots of people, like, if something's broken, they don't go to fix it. They just
then
throw it
also,
away.
yeah, in bigger institutions, like I know where I was doing my postdoc, they sometimes have, where we were having a service where you can bring your material and they can repair it for you on site. So that's also something where you don't toss the material when it's broken, you repair it first, to try to prolong the lifetime of
Yeah.
this. Yeah.
Yeah,
And,
and if I can add something not to forget, also think about furniture, because lab furniture very often is dismantled, is dismissed from certain labs, and the searches on site that were suggested by Julie can actually offer some fantastic opportunities for saving and at the same time for recovering materials that are definitely not meant for trash.
Yeah, totally.
yeah, and repurpose same benches just for another lab or stuff like that, or drawers. And another question I have, because Federico, you mentioned that you use the media but without antibiotics, right? But what about the media with antibiotics? Because lots of labs, including mine, for example, we were always using antibiotics, and I think, you know, most of the labs are using more than one antibiotic in their mediums. So what could be a good solution to repurpose this spending media instead of maybe tossing it down the drain, as many labs are doing?
Thanks for opening this Pandora's box.
Okay.
So no, basically,
service.
no, it's actually a wonderful question. So there are several aspects that we should take into consideration. Antibiotics are a broad class of molecules, and they may actually, let's say, end up being treated in several different ways. We are currently, let's say, focusing mostly on media that does not contain antibiotic, but trust me, in our pipelines and in the project that we are developing, we haven't forgotten about the people who use antibiotic, who are just, let's say, the other side of the coin, let's say. So in this respect, there's multiple aspects to be taken into consideration. Within the reformulation activities, which I cannot disclose in details, there are also some treatments that include, for example, heating the medium, the exhausted cell culture medium. And some antibiotics can be degraded by heating. So in this respect, depending on certain antibiotic types, some of these molecules can actually be processed and end up in the reformulation because they will simply disappear thanks to the reformulation activities. In other cases, there's one additional aspect that should be taken into consideration. That is, some specific microalgae strains, they can actually degrade antibiotics because they are resistant to antibiotics on their own. So in this respect, and this is something that is currently ongoing quite extensively in the lab, we are actually considering specifically some waste media that contains some specific types of antibiotics for these type of treatments. So, and this is part of ongoing work, so I cannot disclose much more. But clearly, what is required is the upstream collection, evaluation, storage. And for example, regarding storage, I didn't spend that much time regarding this, but Julie was mentioning the fact that if you want to do this type of recycling, upcycling, reprocessing, you need space. For us, this is mandatory because we are collecting liquid waste. And one of the biggest problems that we were having at the beginning was related to having cold space for the materials. So the very first thing that we wanted to develop, and we succeeded in this, was strategies to transform the exhausted media into something that can be stored over a long time frame at room temp. So this was step one in our pipelines. And in this respect, obviously, if you work with different antibiotics, that also requires finding the right space and the right workflows to monitor exactly what you have collected and how you are going to reprocess it. But yes, antibiotics are in the pipeline, at least some of them.
Yeah, nice to know. But so in your opinion, like, what would be the best way if you had medium and you didn't have something to do with it? What would be the best way to discard it?
Well, again, I think that it depends on what the medium contains. Probably as of now, the best strategy is if you're not working with a large amount of medium, reprocess it in the autoclave so that you can perform heat destruction of most of the, let's say, dangerous toxic molecules that you have inside. And perhaps, if compatible with the type of medium that you're using, this will not require adding bleach or any other toxic chemicals that will end up polluting the environment. If you have different strategies, such as, for example, microalgae cultures or some other researchers who may focus on bioremediation, consider, let's say, transforming your exhausted cell culture medium into something that can become a source of studies for other people. And this is the option that we're currently taking into consideration, which I encourage to consider in a more extensive way.
We can contact you, and then maybe we can send you our medium, and
if you
can grow microalgae in it.
If you have large volumes, we can definitely think about it.
Amazing, then we can be in contact. So are there any other questions from the audience that we can take? Or something else that we need to discuss? If not, I want to remind you to stay tuned in our social media pages, on our YouTube channels. Interviews are coming. Our special issue of our newsletter for Pride Month is on its way, and you will be able to check it by the end of the month. Of course, we will be in the Matrix Biology European Conference. So if anyone is in there, we can stay in touch and have a drink or two and discuss further. If you have any ideas, please contact us. If you want to be featured in our newsletter, also please stay in touch. Thank you all for your presence here today, and of course thank Julie and Federico for the amazing discussion and all the ideas that they gave us. Hopefully maybe a lot of us are going to implement this in our future, and yeah, thanks a lot for being here, and wish you a nice summer for anyone that has started their vacation already.
Thanks for having us.
Yeah, of course.
Yeah, thanks a lot. Thanks a lot.
Thank you for being here.
Have a good one.
Bye, everyone.
Bye.